Hello, my name is Nicole, and I'll be talking about the conversion of primary cells into immortalized cell lines. These are some benefits to the use of immortalized cells. They are cost-effective and easy to use. They have an infinite lifespan and are great for long-term experiments. They also contain a uniform cell type as they provide pure populations of cells and consistency across samples. The first step involves the direct removal of primary cells from living tissue for in vitro research. These are some of the issues that are associated with primary cell cultures. Their telomeres shorten after every cell division, leading to senescence, eventually hindering the ability to divide after a few cell cycles. For long-term experiments, there are limitations in the reproducibility of new batches of primary cells due to different harvesting conditions. Tumor suppressor genes such as p53 and RB are present in cell cycle signaling. These genes prevent cell proliferation when the potential for DNA damage is higher after multiple cell cycles. Problems can arise when performing research where the goal is for cells to proliferate indefinitely while avoiding batch-to-batch variation while the genotypes and phenotypes remain the same. Immortalized cells can help obtain these conditions for a given experiment. Immortalized cell lines are primary cells that have been altered through the presence of lengthened or stabilized telomeres or the inhibition or knockdown of tumor suppressor genes. Step 1 is the selection of your immortalization reagent. The first option is the modification of telomeres using the human telomerase reverse transcriptase protein. In order to target the telomeres, cDNA coding for the hTERT protein is inserted into primary cells. The TERT protein is the catalytic portion of the telomerase complex that adds simple sequence repeats onto telomeres and compensates for their shortening. This activates telomerase activity. It's simple and causes little change in cell characteristics, but there is a possibility of low immortalization success. Telomerase levels can be tested using Western blot analysis of cancer cell lines. Our second option is targeting tumor suppressor genes. In order to inactivate the tumor suppressor genes, viral oncogenes such as SV40T antigen and HPV E6, E7 are inserted into primary cells for the cell cycle to continue by disrupting p53 and RB pathway. This method takes effect faster, but it can cause change to cells' characteristics. Applying this method alone may not always be successful. In many cell types, a combination of the TERT DNA and the viral oncogenes is required. And step 2 is the selection of your immortalization method. This involves inserting the reagent into a vector through two different methods. Lentiviral transduction is the preferred method, which involves cytoplasmic delivery of the gene of interest and a marker for antibiotic resistance. The antibiotic is added to the culture to select for successfully transduced cells. A range of dilutions of the lentivirus are performed in order to isolate individual populations that have the most ideal level of expression. This will eventually lead to the generation of monoclonal cell lines. Method 2 is plasmid transfection. Although this has a lower success rate, it's a good alternative method for cells that are resistant to the lentiviral transduction. Now, these are some of the challenges that arise, including the need for time-consuming dilution procedures, limited gene delivery methods, possible cytotoxicity, issues with cross-contamination, as well as population mixing. While short tin and repeat profiling can be used to identify cell lines experimentally. However, even with these challenges in mind, immortalized cell lines can exhibit properties including higher proliferation rates, plating efficiency, and saturation density. The establishment of a continuous line of cells can provide an in vitro model for the study of carcinogenesis in human cancers. And these are my references. Thank you for listening to my presentation.